Hi, thanks for coming. I'm going to give a few examples of how we're working with specifically some of the genetic findings in the autism field, taking those back in the preclinical space and trying to apply that for drug discovery purposes. I'm going to give a little more of a conceptual talk, and I hope you find that useful. I'm happy to offline talk about some of the behavioral neuroscience or other types of research going on in the field and talk about examples or approaches that are currently underway to discover new therapeutics for autism spectrum disorders. Yeah. Is that better? Yeah, okay. So first I just want to mention, I'm from Autism Speaks. It's a research and advocacy foundation in the autism space. I hope you've heard of us if you're working within autism research. We were founded in 2005 by Bob and Suzanne Wright, the grandparents of a child with autism. And to date, since that founding, we've invested approximately $200 million in scientific research. So we hope to be a significant influence on science in the field and ultimately on improving quality of life through the discovery of treatments from pharmacology to devices to medical foods, all sorts of aspects of treatment. Some of our major initiatives right now in the science department include an increasing focus on translational research. We have a 10,000 genomes project where we partnered with Beijing Genome Institute ultimately to do whole genome sequencing on 10,000 individuals with autism and really develop a public database as an unprecedented tool for research ultimately. This is a long-term endeavor. It will take quite a while to do whole genome sequencing on 10,000 individuals, let alone to manage those data and make them publicly usable. And we have a number of other initiatives focused on diagnostics and early access to care, and I'm happy to talk about some of those initiatives. But today I'm really going to the content of my talk is more oriented towards our translational research focus. And within translational research, we have a number of mechanisms where we're hoping to influence translation of science into therapeutics. One is a new venture philanthropy affiliate that we've launched to partner with for-profit entities to help facilitate the delivery of novel therapeutics into the marketplace and make them available for the community. We do a lot of targeted projects where we might identify a gap in the field, find investigators to develop a project with, and partner on answering questions through basic research, clinical research, and with a translational focus. We do a lot of traditional investigator-initiated research through RFAs and have a postdoctoral fellowship in translational research, and I'm happy to talk about any of those opportunities if you're interested. So I'm talking about translational research in the same way that we've been discussing it so far in this session, this classic bench-to-bedside approach, and really bridging this gap and bringing clinical findings back into the preclinical space to help influence research at that point, and ultimately with a purpose of reducing risk of failure in late clinical stages. So aligning these two components, and this could be as simple as using neurophysiology approaches in preclinical animal models that have analogs that are usable in the clinic. EEG, for example, could be one. That's not to say you can't use multi-electrode recording arrays in animals and not have relevance in the clinic. You certainly probably could if you start focusing on circuits as opposed to methods. It's not always a linear relationship in that case. I want to mention also an IMI project that's focused on autism, the EU AIMS project. If you haven't heard of the IMI, I encourage you to go to the website if you Google IMI. And if you do that, take a look at the EU AIMS project. It's a really interesting and unprecedented collaboration between pharma and large medical universities in the EU. We're a partner in this as well. In this project, there are work packages ranging from developing in vitro models with a focus on iPS cells, for example, derived from patients with autism that have a genetic variant that is associated with their condition, through to using genetic models of autism-associated genetic variants for discovery of therapeutics into a lot of clinical imaging, back-translating clinical imaging into imaging in animals, and a clinical work package with aims along the lines of things like identifying what types of outcomes measures might be sensitive to detecting deficits or impairments in autism, and then also to treatments. So for the most part of my talk today, I'll do an introduction to the clinical presentation of autism, move into using human genetics, or where we stand right now with our knowledge of human genetics. And in 30 minutes, it will be a brief overview And if I have time, I'll talk a little bit about a symptom domain approach using oxytocin as an example at the end. And this is sort of an RDoC-based or research domain criteria-based perspective. If you have not heard of that, it's been mentioned in previous talks, but also check out the NIMH website and look at the RDoCs approach. It's a very interesting model. So in terms of what autism is, the clinical presentation by DSM-4 criteria, there are 3 core symptoms. These are social impairments, language communication impairments, and repetitive behaviors and restricted interests. And there are multiple adjunct or associated symptoms. Some of these are psychiatric in nature, mood disorders, anxiety-related disorders, OCD. Some are more neurological in nature, seizure disorders, epileptiform EEG phenotypes are not uncommon in autism, and there is a lower threshold for seizure in many individuals with autism. There are, of course, also somatic symptoms, and John Crine did a great job really of introducing the fact that GI symptoms and GI disorders are very prominent in autism. Maybe up to 90% of individuals have some sort of GI tract dysfunction. So it's a very important area, somewhat under-recognized, but of increasing interest. And I'm not really going to touch much on that today. But the DSM-5 came out about a month ago. So what did that do for us? So in the DSM-4, autistic disorder was one of the pervasive developmental disorders. Now it's autism spectrum disorder. It's not within, there's no longer this PDD category that encompasses autism. And autism spectrum disorder subsumed Asperger's syndrome. There's no longer an Asperger's diagnosis. It's subsumed pervasive developmental disorders, not otherwise specified, so that's also not a distinct indication anymore. This is partly driven by the clinical reality of there being autisms and not autism. It's the same in depression or schizophrenia. There are subtypes, and there are multiple subpopulations within autism that may be identifiable either on etiology or symptomology, and for treatment purposes, it may be very important to identify what those subpopulations are. In the DSM-5, we moved from 3 to 2 real core symptoms, bringing the language, communication, and social deficits together into one. It makes a lot of sense. And then also elevating in importance sensory processing differences in autism. These, over the past few years, have become much more highly researched, and their link to repetitive behaviors is a bit more clear. There's also this social communication disorder that has been presented now in the DSM-5. This is individuals with social communication deficits, but do not meet criteria for restricted behaviors and repetitive behaviors and restricted interests might fall into this category. So there are also prominent comorbidities, especially in psychiatric symptomology, and not to read through all of this, but the gist is that nearly half of individuals with autism have any emotional disorder in this category. These encompass multiple types of anxiety and mood disorders. You'll notice that about 30% will have oppositional conduct disorder and also meet criteria for ADHD. In terms of etiology, it's about as heterogeneous as the symptomology, if not more. There are multiple genetic influences, and there's evidence building for some specific environmental influences. In terms of inheritance, old twins or some of the original twin studies indicated a concurrence rate in monozygotic twins of up to 90%. It's probably a little lower than that, maybe 70. But these estimates are probably relatively accurate, and in dizygotic twins, much lower, maybe 10 to 30%. Being male is also a significant risk factor. For our understanding at this point, the ratios are about 4 to 1, male to female. As diagnostic accuracy gets a little better, as physicians are better educated to identifying individuals that are less severely affected, we're finding that this ratio may flatten out a bit. It turns out that there are more females that have a less severe phenotype than there are males, and there could be this a sex protection effect. So some of that sex difference may level out in time. Monogenic disorders, so there are some neurodevelopmental disorders that are caused by single gene mutations that we're very well aware of, including Fragile X syndrome, Rett syndrome, Phelan-McDermott syndrome. These are all caused by mutations in one single gene, and a significant proportion of individuals with those diagnoses also meet criteria for autism or have symptoms that overlap with autism. Unfortunately, advanced parental age is another potential issue. prenatal insults, we've heard a bit about valproate model. Low birth weight, maternal obesity are also potentially significant risk factors for developing autism. In terms of epidemiology, epidemiology is very dependent upon how you do your sampling. Right now, there have not been many studies outside the U.S., and even within the U.S., they do vary a bit in methodology, but the consensus is really where we sit right now at about 1% to 2% of the population if you look across studies. The highest prevalence rate that's been reported was in a study actually that we funded in South Korea, that instead of using retrospective approach and looking at medical records and school records like the CDC has done when they reported early or late last year a 1 in 88 number, this was a much more direct polling of individuals and confirmation of diagnoses. So when you use that type of approach, it looks like we're a little closer to the 2% mark. Another study in the U.S. was conducted with somewhat similar methodology where parents were contacted directly and polled for a confirmed diagnosis. That led to about a 2% prevalence rate indication as well. Again, this is driven by different methodologies. A lot of countries do not have the awareness or diagnostic awareness and accuracy that some other countries have, so this has yet to be fully developed internationally, but right now we sit at the thought that we're at about a 1% to 2% prevalence rate. So what is the status of medicine for autism right now? Well, the standard of care is ABA therapy, so applied behavioral analysis. Individuals that are diagnosed will undergo or can undergo a very robust ABA treatment regimen. But in terms of medicines, there's really not much. 2 antipsychotics have been repositioned for the treatment of irritability symptoms being aggression, tantrums, self-injurious behavior. So essentially these 2 D2 antagonists suppress behavior. Their sedative actions may be the critical component here. That's not to say this is unimportant. Aggressive outbursts in adult can be very threatening. So these have been effective in helping some individuals manage symptoms, in particular in relation to self-injurious behavior. But we're really only treating that subset of symptoms as opposed to this entire symptom picture, and in particular, the core symptoms are really not affected in any way with these treatments. So this has led to a polypharmacy approach where all these various categories of compounds developed for other indications are being used with very little randomized controlled trial evidence to support their use, in particular in this population, in particular in pediatrics in this population, and then on top of that, as an adjunct to applied behavioral analysis. So we know historically from the depression literature that combining medication with psychotherapy can have a more significant effect than either one given alone. In autism, if individuals are in a robust ABA therapy program, it's much more intensive than seeing a psychiatrist once or twice a week. And the idea of combining medication and looking for synergistic effects or optimal treatment has not really been well explored in the clinic. Almost none of these mechanisms have been explored in a double-blind placebo-controlled trial in individuals with autism. There's few exceptions to that, but most of those are still relatively underpowered and not replicated. So where I'll get to now is the gist of where we stand with human genetics and other approaches, and I want to highlight 2 approaches that are currently a focus for identifying novel compounds that might treat the core symptoms of autism. One is starting with human genetics, and the other is using a symptom circuitry domain. Again, this is very much, I'm referring to an RDox approach, research domain criteria approach from the NIMH. One is clearly very disease biology-based, would implicate a very personalized medicine type approach, in some cases maybe an extremely personalized medicine approach where we're developing treatments for individuals with an extremely specific genetic etiology. In the symptom circuitry approach, that's also disease biology-based, but we're really looking at and using our knowledge around symptoms and what we might know about circuitry underlying specific symptoms or cellular abnormalities that might underlie specific symptoms, and then applying that to broader categories of patients. So in terms of the genetics of autism, we've heard a bit in a few other talks at this meeting. Right now, the estimates of genetic causes in terms of who's identified to have one, we think that maybe 20 to 40% of individuals right now have a known genetic cause of autism. This is changing very rapidly as technologies improve. So in the case of de novo single nucleotide variants, for example, as technologies get better for detecting those, and as we apply those technologies to biorepositories, as those biorepositories increase, we're identifying more and more. But at this point, I think there may be maybe 400 to 500 individual genes associated with autism. That's likely to double and more. I mentioned that single gene disorders do contribute a Mendelian inherited component to autism, and chromosomal rearrangements, copy number variations are also very, very common genetic causes in individuals with a genetic cause of or association in autism. Some of the most frequent and most highly penetrant, and by highly penetrant in this case, outside of the monogenic disorders, I'm talking about probably this might account for 1 to 3% of patients, and certainly not all of these individuals with a specific chromosomal abnormality or CNV will develop autism. There's a lot of diversity in where this can go. And if you look at the 15q1113 region, for example, so microduplications and microdeletions can lead to somewhat different disorders. In one case, the maternal deletion of the UB3A gene within that region causes Angelman syndrome. Another mutation within the same region causes Prader-Willi syndrome. These are very different syndromes with a significant number of patients that have autism symptoms, but they are still, you need to keep in mind, they are still very distinct syndromes. They're very distinct from an idiopathic autism phenotype. But some of these same mutations, and it's not specific in terms of which genes might be mutated, but also lead to an autism diagnosis as opposed to a distinct Angelman's or Prader-Willi's syndrome diagnosis. 16p11.2 is the same case. Some individuals with rearrangements within that region meet criteria for autism. Others meet criteria for schizophrenia. If you look at 7q1123, that's particularly interesting in the case because deletions will lead to Williams syndrome, but duplications lead to autism. Williams syndrome is characterized by a jovial personality, hypersocial personality, very socially engaged personality. You have the complete opposite in autism. So within this one chromosomal rearrangement, we have two very different types of mutations leading to two very different phenotypes, but along a very interesting continuum. So that's one particular case where it's, there's some very interesting promise. So I think I've conveyed the complexity of the phenotype-genotype relationships. The concept that I like to convey is that there's a many-to-one and a one-to-many relationship. By many-to-one, I mean that you can have mutations across multiple genes leading to the same phenotype, individual mutations in different genes leading to the same phenotype, and one-to-many, that the same mutation or a mutation in the same gene can lead to very different phenotypes. So the picture is relatively complex. And I would argue that's not all that different from what we've heard thus far, in particular with what we're learning about schizophrenia genetics. So how do we make sense of this? Well, in one case, there's a significant focus on networks. So do these genes form functional groups that we can cluster together that give us some clue as to where to look for a biological starting point? And there are clusters emerging from some types of analysis. Brain gene expression using postmortem tissue has revealed differences in different clusters, in particular in clusters of neuronal function and immune function. In one study, showed downregulation of neuronal clustering, and these were for genes associated with axon guidance, for example, dendritic arborization, cytoskeletal trafficking, and vesicular movement. It also revealed an upregulation of some immune markers. These included markers of astrocytes, markers of microglial activation, and other inflammation markers. De novo mutations have also been shown to code for proteins that are highly interactive and overlap in many cases with previously associated CNVs, recurrent CNVs that have been associated with autism. And overall, we're seeing an implication of dendritic organization, axon guidance, and recent evidence for beta-catenin chromatin remodeling from the de novo literature, and looking for how these genes might cluster and are there pathways to focus on. This is still we're still pretty far away from identifying specific targets that a drug company might say, let's jump on and pursue a program. But in terms of basic research and honing in on some specific functional classes in a very complex field, this is definitely progress. We're also learning a lot by studying monogenic syndromic neurodevelopmental disorders like Fragile X syndrome, Rett syndrome, Phelan-McDermott syndrome, and others, Angelman's. In this diagram from Huda Zogby's lab, they really just indicate where some of these genes and their protein products are located, and that they tend to localize to the postsynaptic space. And in this diagram, get my pointer. You have MECP2, FMRP, and other, mTOR and tuberous sclerosis complex. All of these localizing somewhat to dendritic spaces and downstream signaling of NMDA, metabotropic glutamate receptors, having some convergence on signaling pathways that might be mineable for further targets. And if you expand this out more broadly to include a lot of the other the GWAS results and associations and linkage studies in autism, there is a significant clustering of some genes that implicate synapse biology. So many of these genes are located in postsynaptic signaling spaces. Neurexins and neuroligands are very important for developing synaptic contacts. Shank and Homer proteins for developing scaffolds between NMDA and some mGlu receptors, for example. There are genes for calcium channels and sodium channels that also regulate neurotransmission through glutamatergic synapses that have been implicated by genetics in autism. And the field has been very prolific in developing animal models of many of these variants. There are multiple mouse models and an increasing number of rat models in this space. One rationale for developing rat models is because of their behavioral repertoire differing somewhat from mice. In particular, rats show juvenile play behavior. Their social behavior seem to be somewhat more complex in some specific cases than can be assessed in mouse. They're also larger. This is, of course, makes them slightly more amenable to biosampling, repeated biosampling, somewhat potentially to some electrophysiology approaches. And then also in the pharmaceutical industry, where rats are most commonly used in very early or pre-IND safety and tox studies, having your efficacy work and follow through safety and tox work in the same species might streamline the process of developing a target to some extent. So I want to move on to one example now of how genetics have been used, and I'm going to use this example of the promise of the post-genomic era. And if you're in the autism field, you're probably very familiar with the mGlu-R5 or glutamate theory of Fragile X syndrome, and some of the work from Mark Baer and many others in the Fragile X field. They really assessed this approach, starting with identifying psychiatric disease, identifying a genetic cause, leading to making preclinical animal models, assessing mechanism in those models, and turning that back around to developing specific molecules to target receptors within the biology that they've discovered using models of the human genetics. If you're not familiar with Fragile X syndrome, Fragile X is the most predominant genetic cause of intellectual disability. It's also the most prominent single genetic cause of autism. It is caused by a mutation in the FMR1 gene, the Fragile X mental retardation 1 gene on the X chromosome. This causes transcriptional silencing of FMR1 and prevents the expression of FMRP. FMRP is a translational repressor. And So within this field, by identifying FMR1 gene as the single gene cause and then developing animal models of this, they've been able to demonstrate in some classic supporting data for this theory differences in activity. So in this case, they're looking at protein synthesis in FMR1 knockout and wild-type mice. They've also done a significant assessment of protein expression and dendrite morphology. So finding differences in this case in wild-type mice. This is just dendritic spine morphology, and in the knockouts, you see a much more dense or increased density of spines. These spines, if you look at them a little more closely, you also see that they're slightly more immature. There's more philopodia in these animals at this particular age than in the wild types. If you cross these with animals that are heterozygous for mGluR5, so express about 50% of normal mGluR5 receptors, it corrects that particular morphological abnormality. It also corrects protein expression in the hippocampus. Again, the same figure, so wild-type knockout, and then in the cross, you see correction. This was the foundational data supporting or generate an mGluR hypothesis of Fragile X syndrome. FMRP is downstream of mGluR receptors, driving activity through mGluR5, does regulate FMRP. FMRP in turn regulates protein synthesis. So this led to the theory. The theory then led to a resurgence in mGluR5 receptor research, and in particular, and other targets within the mGluR5 signaling pathway. Multiple molecules have been pushed into the clinic, and where we stand right now is really that Roche and Novartis are in relatively late-stage trials with selective mGluR5 negative allosteric modulators for the treatment of Fragile X. So this has been a relatively successful and interesting example of starting with understanding a genetic cause of a very specific psychiatric subpopulation and moving through animal models into basic neurobiology, developing a mechanistic theory, and taking that mechanistic theory to identify specific molecules and taking that into the clinic. These results are, it's actually very encouraging. Both Roche and Novartis have continued to expand their clinical trial program. Very little information has seeped out into the public space from their phase 2 trials. Novartis is in phase 3. They are moving from adult populations into pediatric populations at Roche and Novartis, which is also they're very encouraging signs. So keep your fingers crossed. One other molecule that's relatively late stage was arbaclofen. It's a GABA B agonist. It's an enantiomer of baclofen. That had also been through phase 2 trials in Fragile X syndrome and in autism. And if you follow the health news generally, this was a bust recently in Fragile X and in autism. Not so much for lack of efficacy potentially, but for lack of efficacy on endpoints that may not that were relatively experimental. So they're really blazing a trail, identifying specific outcome measures, and running trials using those outcome measures, and really informing us of whether or not those are the appropriate outcome measures. And this GABA B mechanism, the rationale is really that it's the other side of the excitatory inhibitory hypothesis underlying some of the most recent autism neurobiology research. So in the case of Fragile X and mGluR5, this story has been relatively clear. It's certainly not simple. There are lots of complexities along the way, and we still don't actually know the results of these large-scale clinical trials. Those will, of course, be the ultimate test. But in terms of moving from a specific genetic etiology into the clinic, it's a great story thus far. This won't always be the case. From just one other example in the autism space, SHANK2 gene mutations have been associated with autism and intellectual disability. 2 mouse models have been made of mutations within the SHANK2 gene, one encompassing exon 7, one encompassing exon 6 and 7. It doesn't sound like there'd be much difference, Yet when, I just use the example here of measuring LTP in these animals, in one of them, NMDA receptor-mediated LTP was hypersensitive. In another one, there was actually a deficit in NMDA receptor-driven LTP. This is potentially due to methodological differences. Maybe it reflects, though, the diversity of what can of molecular mutations in a specific gene and how that can alter signaling in different ways. It's not simple, but it's also still fruitful to go down this path and ultimately to collaborate and find replication and clarify the mechanisms. So if I still have a minute, I'm going to quickly touch on the symptom circuitry or RDoC domain approach and how we might look at oxytocin as one example of this within the autism space. We heard quite a bit of background on oxytocin thus far in the meeting, so I'll skip some of the historical literature implicating oxytocin as a mediator of social behavior. It is across species that some specific effects have been shown, so cumulative evidence does support that there are pro-social benefits of oxytocin in different populations, and certainly in some animal models. I use these two examples on the right to relate to the RDoC approach because they both implicate the amygdala. So one is a human imaging study showing that intranasal oxytocin reduces activity in the amygdala. So if you look there, you see the amygdala, you see activity bilaterally in the amygdala in those two spots. If you look in this graph, you do not. These are the intranasally treated individuals with oxytocin. These are young women who have not had children, and this is their response to infant laughter. Oxytocin reduced the amygdala activation in response to infant laughter. The interpretation is that oxytocin acted within the amygdala to potentially attenuate regions that might cause aversion to different stimuli. And this is just one example. There are multiple examples of oxytocin acting within the amygdala and regulating activity. I thought that was one particularly interesting study by potentially reducing activity in brain regions regulating maybe aversive reactions. You might be increasing the reward salience and facilitating greater maternal behavior. That's the interpretation anyway there. The point I want to make is that it's another demonstration of oxytocin regulating activity in the amygdala. This is a diagram from some very elegant preclinical work done in Ron Stoop's lab, where they have very carefully demonstrated that oxytocin specifically regulates a subpopulation of neurons within the amygdala that project to the periaqueductal gray and regulate freezing behavior. That is in contrast to another subpopulation of neurons projecting to the dorsal vagal complex that regulates heart rate in response to fear, to foot shock, essentially. So this is a fear conditioning-based assay where they have very finely dissected how oxytocin is working in a microcircuit within the amygdala and projecting out to regulate some components of an emotional behavioral response. Overall, the main point I want to convey is that this brings together across species an implication for an amygdala-centered circuit. And again, it's not just these two studies. I find these to be two interesting studies. Ron Stoop's work, this is actually a compilation of a few very elegant papers. And again, just building this case for a circuit-based approach to treating some symptoms that might span disorders. So I'll conclude. There's clearly a profound unmet need for medications to treat core symptoms in autism. Autism genetics does suffer, maybe you'd say, from high locus heterogeneity. It's certainly more complex than I think we all hoped it would become, but there is growing evidence for some clustering, and as some methodologies evolve, hopefully we'll identify specific networks and clusters that lead to targets that we can pursue. Human genetics and symptom circuitry domains, they're certainly tractable, and I think honestly embracing some of this risk is really the only way forward. I don't think we can shy away from the complexity of these genetics. If we do, we're going to have to shy away from schizophrenia. We're going to have to shy away from mood disorders. We're going to have to probably do very little in the CNS space. It might be complex, but we're all scientists, and I think you can deal with this, and it's really investing in this and embracing it is the best way forward. And some companies are really pioneering this approach, and and ongoing clinical trials are going to be a great test of both of these symptom-based and human genetics approaches. So I'd say think beyond the brain and genes as well. We heard from John Cryan about GI tract research, and that's certainly a field wrought with opportunity. And we need to consider gene-gene interactions and gene-environment interactions. Obviously, neither one operates in isolation. Not to add to the further complexity of it, but just to hopefully stimulate and reinforce your interests in this if you're already exploring those areas. So that's it. If you want to contact me, please feel free to send me an email or talk to me after the meeting. Again, I'm happy to talk about other work going on in the field and some of the programs we have at Autism Speaks.